In this video, I'm going to be talking about Psmac. So pSMAC, it's basically tumors hate because it enables everything that immune cells have to do in order to defeat tumors and to reduce cancer cell numbers. Okay, now why do tumors hate Psmac formation? So Psmac is the killing ring. It's made of anchors actin, also transmits force, also granule polarization, perforin release. So basically, perforin is it's like a tube that attaches to a cancer cell, and then the granzyme B goes in through the tube and then makes the cancer cell go through apoptosis. Also, Psmac enables death receptor clustering, or Fas-facile-mediated killing. So basically, Fas-facile is mediated apoptosis in tumors. So there's Fas-CD95, which is the death receptor on cell surfaces. Fas ligand expressed by activated NK cells leads to caspase cascade activation, apoptosis of tumors. So apoptosis is programmed cell death. Caspases are also activated and modulated by potassium. So depending on potassium levels being high or low has a different effect on caspases. Also, potassium needs magnesium to work, and then but magnesium needs vitamin B6 to work as well. So without those two, potassium cannot modulate the caspases levels that affects the apoptosis. Now, once the Psmac is formed, and this is why tumors hate the Psmac, is the outcome against the tumor is sustained immune contact, effective cytotoxicity, and immunological memory priming. But tumors, they use Acetyl-CoA, which is caused by high sugar levels combined with high amino acid levels, or protein, especially leucine. And tumors, they also use Acetyl-CoA to force synapse slippage. So basically, the synapse of the immune cells, it's like a bridge, an anchored bridge that the immune cells need to be able to target and attack cancer cells. But the tumor will try to stop this from happening with Acetyl-CoA. Okay, another trick that tumors use against immune cells is that they cause immune cells to burn through and to waste ATP, but gaining no traction. This is due to the actin polymerization becoming retrograde without anchoring. Now, actin polymerization is important for the immune synapse formation, or like the bridge, the anchoring bridge that the immune cells have to stand on. Without that, the immune cells, they literally slip off of the surface, since the actin polymerization is disabled. That's supposed to be to strengthen the surface, but instead tumors degrade that. So what that ends up doing, which is the degradation, is premature disengagement. So the immune cells stop attacking the cancer cells. Exhaustion signaling, which is not good, because T cells and immune cells, they have a lot of different exhaustion markers, such as TIGIT, CD, etc. Also, failed killing despite recognition. So even if immune cells see the let's say the targeted cancer cell, it doesn't attack with, like, let's say granzyme B, perforin, or Fas-facile-mediated apoptosis. Now, this next scenario is actually perfect from the tumor's perspective, since the tumor hates the Psmac so much. This is also why it's a good idea to avoid excessive high glucose with excessive protein or amino acids, especially leucine. And due to the tumor and using Acetyl-CoA, it causes slipping synapses, and uh, the immune cells, they cannot have slipping synapses. It has to be like anchored surfaces caused by proper actin polymerization. Only if there isn't acetyl-CoA from too much glucose and too much protein together with glucose. So if that is the case of acetyl-CoA, the slipping synapses causes short dwell time, incomplete signaling, no lethal hit delivery. That means that high sugar combined with protein does not allow or prevents the immune cells from literally attacking cancer cells, such as with granzyme B perforin or other ways like FASL, and increased immune exhaustion markers, as I explained already. Okay, so basically the tumor knows how to make the immune cell to not stay, or the tumor won't allow the immune cell to stay. So the immune cell finds the surface or finds the cancer cell or the tumor, but the immune cell can't stay. Now, there are some ways to destabilize the tumor's membrane advantage. And one of those main dietary compounds and substrates is omega-3 fatty acids, so found in a fish, sardines especially, or even you could just buy supplements. But the critical component and effect of omega-3s is that it leads to mild glycolytic throttling by limiting citrate overflow. So this dealing with the citrate flow by omega-3 is critical because acetyl-CoA itself does not cross the mitochondrial membrane. Since tumors, they inflict reactive oxygen species onto the mitochondrial membrane to weaken it. Because if tumors cannot get the acetyl-CoA to cross over to the mitochondrial membrane, it'll just generate citrate from acetyl-CoA, from high glucose, high protein, with the citrate carrier that tumors express, which is SLC25A1. So once the tumor has done all those steps, it leads to protein acetylation, or basically, because it's happening, the protein is acetylated because it's being combined with high glucose, which is going to favor the tumor and avoiding immune attack. Tumors also aggressively upregulate SLC25A1 so that it can deal with the citrate. Now, another thing here that tumors do in order to try to stop the Psmac activation, because the tumors really hate it, it can really it's basically like the end of the tumor if the Psmac succeeds in forming. This also explains why the lysosomal membrane cannot be peroxidized and why it must be taken care of with, let's say, resveratrol, magnesium. So the tumor will try to load the lysosome full of cholesterol. And the reason it does that is that the lysosomal membrane being full of cholesterol will destabilize the lysosome. And then the lysosome will not be able to deal with and degrade the exosomes, and then the exosomes go over to the tumor. So the tumor wants to receive the exosomes, because if it does, then that'll make it more difficult for the Psmac to form, or at least harder for the Psmac to attack the tumors. And once the tumors succeed at getting the exosomes, and the tumors go through the exosome cargo, the tumors are able to do all kinds of different things to destabilize the immune system, and possibly the SMAC as the Psmac as well, which is actually beyond the scope of this video. So that's it for this video. Like, comment, share, subscribe, and I'll see you in the next video.